All right, everybody. Welcome back. This video is going to be for chapter 37, which is soil and plant nutrition. So the first thing that sort of just like the overall scope of this chapter is examining how soil and plants interact. So the first thing is what is coming from the soil. Well, let's break down a plant, right? First thing I want to mention is the biomass of a plant. So there's a misconception that plants gain biomass from the soil, and that is not the case. Plant biomass is very, very primarily going to come from atmospheric CO2. It's also going to gain a lot of biomass from water, actually, from these hydrogens and oxygen as well. So atmospheric CO2, remember, that is going to be what's being taken in by the leaves. So it doesn't have anything to do with the roots. And just a note about like why that sort of lines up is carbon, we know, is sort of the building block of organic compounds and therefore biomass. So that's the first thing. Plant biomass comes from atmospheric CO2. That's very likely going to be a test question. So the reason we know this was a guy named Van Helmont, which is a pretty cool name. The Van Helmont experiment was really simple. He basically measured the amount of soil that he was planting a little tree into. Let's say there were 10 kilograms of soil and 1 kilogram sapling. And then he watered it, regularly for 5 years. And he waited and waited. And after 5 years, the tree had grown a lot. Let's say that the tree grew to be 90 kilograms. But the soil only lost a very, very negligible amount of mass. It only went from 10 kilograms down to like 9.99 kilograms of soil. So really small difference. So that means that the growth of the tree, because the tree gained a lot of mass, wasn't coming from the soil. What else could it have been coming from? The atmospheric CO2 going in and the water that he was putting in. But CO2 is the main contributor to biomass. And we'll see why in just a second. So what comes from soil? What is the point of soil? Why do we need to plant things in soil? Well, it's because we need minerals and nutrients. And then, of course, we also do need water, which soil can hold water for us. So the three sort of like most known, I would say, nutrients that are going to come from soil specifically are going to be nitrogen, phosphorus, and potassium. And each of these is going to have a unique role. But the first thing I just want to point out is sort of the percentage of biomass that these three minerals make up. Because remember, I said most of the biomass is going to be coming from CO2 and H2O. So nitrogen really only accounts for about 1.5% of the biomass. Phosphorus is a whopping 0.2%. And then potassium is probably about 1% of the biomass. So what do each of these do? Why do we need these minerals and these nutrients? Well, for nitrogen, it's going to be an RNA or a DNA building block. And if you remember from first term, the nitrogenous bases, remember, which were those, nitrogenous bases, which was adenine, thymine, cytosine, and guanine. You need nitrogen to make those. The other big thing for nitrogen is it's a major component in chlorophyll, which was the photosynthetic material. So you need it to build RNA and DNA, which is obviously super important, and then you need it for chlorophyll. Phosphorus is also going to be RNA and DNA, because again, if you remember from first term, RNA and DNA have a phosphate backbone. So that's going to be the phosphate backbone, which again, super important. And then the other really, really important thing that phosphorus does is ATP. Remember, the P in ATP is phosphate. It's adenosine triphosphate. So you need phosphorus to make phosphates. So ATP is obviously running all of your cellular processes. So potassium, on the other hand, is going to be a little bit different. It's going to mostly be a regulator of water flow. And you may remember from last chapter that I talked about guard cells swelling up and guard cells becoming flaccid to open and close stomata. Potassium was one of the ions that was actually going into the guard cells to draw in water to open those stomata. So guard cells is a great example for potassium. All right, so we've named 3 elements. We've talked about carbon, oxygen, and hydrogen too, so we've kind of named 6 elements that plants need. In total, there are actually 17 elements that plants need. So we are gonna go through all of them. So oxygen is going to make up 45% of the biomass of the plant. And oxygen is obviously going to be very important for cellular respiration. Remember, oxygen acts as the final electron acceptor in the electron transport chain. So you need, plants do cellular respiration too. A lot of people just think photosynthesis when they think plants, but remember, they're also undergoing cellular respiration, so they really need oxygen. So that's a big role for oxygen. Carbon, on the other hand, like we said, well, first of all, carbon is also about 45%, and that is going to be a big role in photosynthesis as a reactant, because remember, CO2 is necessary for photosynthesis. And then it is also going to be an organic building block, like we said up there. And then hydrogen is again going to be, well, first of all, hydrogen much less. We're dropping down to 6%, which is not much. So hydrogen is about 6% of the biomass, and hydrogen is going to be really important as an organic building block. Again, you can kind of think of like any kind of lipids are going to be carbon hydrogen chains, or hydrocarbon chains rather. And then hydrogen is also going to be a big player in water flow and membrane potential, which is sort of the electrical or like the electrochemical regulation. Nitrogen, phosphorus, and potassium, we already kind of talked about up here, so you can review those if you'd like. Next we have calcium. So calcium is going to be only a very small amount, only about 0.5% of the biomass of the cell. It's very important for the cell wall and for signal transduction. Next up we have magnesium. Magnesium is only about 0.2% of the biomass of the plant. Magnesium's biggest role is again going to be it's a component of chlorophyll. And then sulfur is only 0.1% of the biomass of the plant, and it's going to be really important in the electron transport chain proteins. And just proteins in general. A lot of them will have sulfur as part of their structure, molecular structure. So I've sort of cut it off here. Everything we've talked about up until this point is a macronutrient. Macro meaning large, so these are the nutrients that we consider plants to need a larger amount of. And I know 0.1% is pretty small, but it is going to get a lot smaller when we talk about this next group, which is the micronutrients. So micronutrients, the first one is chlorine, which is only 0.01% of the biomass. It's important for splitting of water in photosynthesis. And then it's also going to help with water balance in the same way potassium did. It's going to go into cells to draw in water sometimes. So next up is iron, which is also going to be about 0.01% of the plant's biomass. Iron is another chlorophyll component, and it's also very important in ferredoxin. And ferredoxin is part of photosynthesis. It's an electron carrier in light reactions of photosynthesis, if you remember light reactions. If not, try to just remember chlorophyll component. Maybe write that down for the note sheet. Next up is manganese, which is 0.005%. So we're getting really minuscule here. Manganese is part of photosystem 2 in light reactions. That's pretty much it for that one. Zinc is down to 0.002% of the biomass. Zinc is helpful in auxin synthesis. Auxin being a primary growth hormone for plants that we talked about last chapter. Boron is also going to be 0.002%, and boron is mostly going to be a cell wall component. And it's also really important in pollen. Next up, we have copper. Copper drops down to 0.0006%. And it's important for xylem function, so transport of water up the chute. Nickel drops down to a very, very tiny 0.00001% of the biomass, and it's for nitrogen metabolism. So help breaking down nitrogen sort of into things that are usable and, able to be used in DNA, RNA. And the last one is molybdenum. Molybdenum is also 0.00001% of biomass, and it is going to help reduce nitrogen. And when I say reduce nitrogen, I mean add electrons, not reduce the amount. So these are the micronutrients. You do not need to know the relative percentages, but you do need to know sort of what's more abundant and less abundant and some of the major functions. She won't ask you where is manganese found, but she might ask you what's more abundant, calcium or manganese. She'll never ask you to compare a micronutrient to another micronutrient. She might ask you, what is the most abundant, and you would say oxygen and carbon. But this is a good thing maybe to jot down in the notes, just sort of the order of abundance and what is macro versus micro. So now that we've looked at all these sort of essential elements, we're going to start talking about soil. So first off is, how does soil form? So soil always starts with solid rock, and then maybe the wind blows that rock a whole bunch, and it shoots off tiny little particles, and these tiny particles can accumulate, and then the rock is a little bit smaller. Then maybe we have a heavy rain or some flooding, and that breaks the rock down just a little bit more, and we get even more particles. Now we have even smaller rocks. Maybe a freezing and thawing occurs and cracks some of the rocks into, you guessed it, smaller rocks, and maybe puts off a few particles. And then maybe those smaller rocks get plants in them, and those plant roots cause them to crack and crumble a little bit. Then we have even smaller rocks and more particles. And then maybe animals are trampling over the ground and the rock, and that is going to make even more tiny particles and a smaller rock. All of these little particles that we have accumulated from these various weathering processes, this is called weathering, all these tiny particles are eventually going to combine to make soil. So sort of the anatomy of soil, they break it down into 3 layers. It's kind of 4 layers, but we'll call it 3. The first one, they're very easy. They are horizon A is the first one. Next up, we have horizon B. And then you can probably guess what the last one is. Yep, you guessed it. It's horizon F. I'm just kidding. It's horizon C. It's ABC. It's really easy to remember. Horizon A is where most of the stuff is going to be going on. Horizon A is also referred to as the topsoil. So the topsoil has another tiny little layer in it, and I've kind of denoted it here, and then we're going to blow it up and talk about it more. This is called the humus. It's not hummus, I don't think. Hummus would have 2 M's and is delicious to dip pita bread into. But the humus is the top layer of horizon A, or the top layer of the topsoil. So if we blow that up and look at what's going on in here, the humus is where we are going to have roots. You're going to have organisms. That says organisms, I'm writing very poorly. Like worms, for example. You're going to have all kinds of bacteria Oh my gosh, that just got really bad. Sorry. Bacteria. You're going to have more roots. You know, you might have little ants even, you know, even more organisms. You could have protists or proteasomes or something. So it's where living organisms are. And then the other major component of this humus is decaying organic matter. So that's dead animal, dead leaves, dead grass, dead bacteria, dead everything that are breaking down into this very nutrient-rich sort of soup, if you will. You can think of the decaying organic matter as the broth of your soup, and then everything else here is your chicken and your noodle and your veggies. So that's horizon, that's the humus. That's the top layer of plants. That's where most of the roots are, and that is part of the top soil. So topsoil is very loose, and then we're gonna, as we move down, get more and more packed. And then down here is basically solid rock. Horizon B is just an intermediate. So that's sort of the anatomy of soil. Next up, we're going to talk about the types of soil. So there are 3 types of soil in general. There might be more, but for this class, there's 3. You have sand, clay, and organic matter. You should all kind of have an idea of what sand, clay, and organic matter means, but if you've never been to the beach, sand is sort of these large granules. I should have written that down too. These are large. And when we say large granules, we mean for soil purposes. They're actually super tiny, right? But they're large granules. They're very dry. So in sand particles, and I should mention this, these are all particle types. Anyway, sand particles are large granules. They're very dry, so there's low water availability. There's also pretty low nutrient availability. You have very high O2 though, and you have a high O2 because of the large granules. There's more space between each granule because they're bigger, so they can't really pack in as tight. Because like here you have an empty space that could be oxygen, you know? Here's empty space that could be oxygen, whereas if we have just a bunch of tiny particles, there's not as much empty space. And that large granule structure, that sort of loose structure, is also going to allow for high root penetration, meaning plant roots will have a very easy time planting in sand. They don't have to exert much force to drive their roots in. Sand carries a neutral charge. That'll be important on the next page, but we will cross that bridge when we get to it. Just know for now it's neutral. The next type is clay. So clay is going to be really, really dense, but it's also going to have very high water. I should also mention clay is dense because the individual particles are able to saturate more with water, which increases the weight, which squishes them together better in a sense. And that's why when you have a block of clay, it's kind of solid. When you have a block of wet clay, I should say. But when you dry that clay out, you can smash it with a hammer and it crumbles so easily. So clay is very dense. It has very high water availability. It has very high nutrients, and I'll talk about why in just a second. But it has very low O2 because oxygen doesn't have, if you have a block of clay that's essentially solid, where am I going to be able to put oxygen? It's too tightly packed, which is also going to lead to low root penetration. So it's hard for something to plant into clay because its roots have to really drive in hard. And the reason that clay has high nutrients associated with it is actually because it carries a negative charge. And because it carries a negative charge, it can attract cations, which are positively charged ions like potassium or calcium that then sit with the clay. And organic matter is the same charge. Organic matter is also going to be negative charge, so we already know it's going to have high nutrients. Organic matter is also going to have high water. It's going to be able to have high amounts of O2, and high root penetration, because it's sort of loose and crumbly, but it's able to be saturated with water very well when you have decaying organic matter. And it carries a slight negative charge typically, which is going to allow nutrients to sort of stick with it a little bit. High root penetration, again, it's kind of loose but can still saturate, so roots can still penetrate. And that's why organic matter is the main component of that humus. It's really, really good at everything, kind of. So those are 3 particle types. We have sand, clay, and organic matter. Moving right along, I wanted to talk about the ion distribution in soil. So right here, I just have a sort of just a cross-section, if you will, of some topsoil. And in this topsoil, I have got these sort of long-looking things are going to be clay particles. So this is a clay particle, this is a clay particle. These blank, sort of large circles are sand particles. And then these sort of funky shaped, misshapen ones are going to be organic matter. So you can see that the organic matter and the clay particles have negative interior charges. And what that is going to do is attract cations. So cations, again, like calcium, which has a 2+ charge, and potassium, which carries a positive charge, are going to attract to these soil particles. And then a root hair, let's say, and we'll get into this later, a root hair that's right here is going to benefit from that because it's going to be right around these particles that it wants in it. So the other thing that we need from the soil, though, is going to be the anions. And just to clarify real quick, the empty space surrounding all of these particles is actually water. So all of this is water. And these particles sort of sit in the water. And obviously this is very watery soil right here because I only have like 10 particles in this whole thing of water. It's just for the sake of understanding though. But this water is also going to be able to hold a bunch of anions. Remember I said cations are positive charged ions and anions are negatively charged ions. That's why there are these free-floating negatives. So this is going to be things like phosphate, which is PO4 minus, or like nitrite, which is NO3 minus, or chloride, which carries a negative charge. And these are going to stay distributed in the water specifically because of hydrogen bonding. So I have, to really like just blow what's happening here up, I have my water, which remember is H2O. So here are my H's and here's an O. These negatively charged particles are going to interact with these slight positive charges of hydrogen. And remember, that's hydrogen bonding. So that's why the anions stay distributed in the water. So the next influence, we talked about ion distribution, and we'll get into absorption in a little bit. But the next thing that I need to talk about is going to be pH. So pH scale is a measure of acidity. So you have 2 types of soils. You have acidic soil and you have alkaline soil. Alkaline is just another word for basic. So acidic soil is going to be very prevalent in forests that have a lot of dead trees or a lot of trees that fall down, because those trees are going to fall down, decompose into a bunch of organic matter. And that organic matter is going to be able to express itself as things like carbonic acid, or phosphoric acid, or nitric acid, which is going to lower the pH of the soil, and when we lower pH, that's more acidic. So we have this acidic soil that's rich in these nutrients. Alkaline soil is going to be more common when there's limestone-rich areas. So limestone is very marine, I think of it as. So like you have like marine shelves or tropical shelves, coral reefs, like the Caribbean, Southeast Asia, places like that, the Mediterranean. And this is going to carry a whole bunch of positively charged things that are going to increase the pH and make it more basic. And that's things like calcium, sodium, magnesium. So that's the effect of pH on soil. We have acidic soil and alkaline soil. So we've gone through pH's influence and we've gone through the influence of ion distribution. So now let's talk about getting nutrients into our root hairs. Remember, root hairs are going to be sort of off of the larger roots. So here we have a root and it might have a bunch of root hairs. We're going to focus on this root hair. So remember up here we had this negatively charged organic matter. So I've moved that down here. Organic matter, negative charge. That negative charge is going to attract these cations like calcium 2+ or potassium plus. So calcium ions and potassium ions. What's going to be able to happen now though is hydrogen, which is sort of protons, I should say, which is H+, which in an aqueous solution can be pretty abundant. Protons are going to be able to sort of switch places with these cations because they have a positive charge too. So when we do that, we go over here and now the hydrogen is bonding and these two cations are free to either go into the root hair or be washed away via water flow. So they could either be washed away from the water flow or they could be absorbed into the root hair and utilized by the plant. That's called cation exchange. So the mechanism of uptake, as you can see, we're getting a little molecular now. So this is going to be pretty classic. We've seen all of these transporters before, but let's go through it again anyway. So let's say that I have some calcium ions out here that I would like to absorb into my root hair. And let's also say that I have some chloride ions that I'd like to absorb into my root hair. How are we going to be able to do this? Well, it is going to be via our good friend, the proton pump, which is an ATPase. So if it's an ATPase, remember that means we are using energy. So this is active transport. So what's going to happen here is I'm going to have some hydrogen ions, some protons, H+, in my root hair cell. So this is the inside of the cell. This is the soil. What's going to happen is I'm going to take ATP and I'm going to break that third bond to ADP. And hopefully you remember when we do that, it releases energy. And I'm going to use that energy from breaking this ATP to start pumping these protons out of the cell. And remember, this is against their concentration gradient. I'm creating a concentration gradient which requires energy because the protons want to stay in the cell, but I'm using ATP to push them out. And that is going to mean eventually I'm going to get a really big buildup of protons out here. And that is going to do two things that are going to help us drive stuff in. The first thing that I want to talk about is the charge on the inside of the cell. And we're not going to get into voltages or anything like that, but I just want you to think. If the inside of the cell, let's say, was at 0, and 0, I don't care about units right now. If I was at 0 and I removed these positively charged protons, so I'm removing positives, what's that going to do to the charge on the inside of the cell? It's going to decrease it. So we are now going to go to like a -5 charge, let's say. This is an irrelevant number. We're just, for sake of, the example, we have reduced the charge. So now the inside of my cell is negative because I have taken away positively charged things. So we're going to have something like a calcium channel, which is going to allow positively charged ions, which were cations, to flow through this channel to this negative area. Because remember, positive is attracted to negative. So this negative interior is going to passively allow this calcium to flow down its electrochemical gradient. can't read that super well, but it says electrochemical gradient. It's going to passively flow, and we are going to get our calcium ions into the cell. All right, great. I've got my calcium cations now that I wanted for my plant. Now I need to get these chloride ions, and that is going to pose a little bit of a problem because remember, the work of this proton pump over here made the inside of the cell negative. These chloride ions do not want to go through here anymore. They don't want to because a negative is not going to like a negative. It wants to repel the negative. So what we are going to have to do is use the other aspect of this proton pump. So this proton pump created this electrical gradient and made the inside negative, but we also have a super high concentration of protons outside the cell now. It's very, very high. So a hydrogen ion or a proton, sorry, I'll just call them protons, these protons outside the cell really, really want to flow back into the cell very badly. So this channel right here is going to allow that, and this is going to be called the proton chloride cotransporter. And remember, we are harnessing the chemical gradient created by primary active transport. That makes this secondary active. I can't write when I'm close to the spiral of the notebook, but that says secondary active transport. So what's going to happen is this proton that really wants to go into the cell is going to do that. It is going to passively go into the cell, back into the cell, because it wants to go down this chemical gradient that we've created with the proton pump. And then one of these chlorides is just going to hitch a ride, because the act of this proton moving from an area of high proton concentration to an area of low proton concentration is going to generate energy, and that energy will be absorbed by these chloride ions to hitch a ride into the cell. And now we also have our anions, or our negatively charged ions in the cell. So that's one mechanism to get ions into the cell. The next one is actually through fungus. So fungus, there's a fungus called mycorrhizal fungi, whatever this word is. I can't say it. Mycorrhizal, something like that. And they have a mutualistic relationship with plants. And mutualistic means both benefit. So this fungi is going to attach to root cells and sort of penetrate into the root. So it's, if I have like a root cell here, this fungi is going to be able to actually go into the cell a little bit and sort of anchor in there. And when it anchors in there, it's going to provide a channel for ions to just go right into the root cell. And in return, it's going to take some of the sugar that plants produce from photosynthesis. So the fungi is getting sugar and the plant is getting nutrients. All right, so the next thing we're going to talk about is ion exclusion. So there are some ions that are actually bad to the plant and can even poison and kill the plant. This could be ions like cadmium or lead or some of these toxic metals, or if you just have too much of a micronutrient like zinc or nickel, that can also poison the plants. So there are two types of exclusions. There's passive exclusion and active exclusion. Passive exclusion is really easy. It just means the plant basically going, okay, if I take in lead, it's going to kill me. So I'm just not going to put any lead channels in my roots because I don't want lead in. So I'm just not going to have a lead channel, and then lead can't kill me. Or it could also be the plant sort of, I say the plant going, I'm anthropomorphizing it, but I don't care. I only need a little bit of zinc in this plant. So what I'm gonna do is just put a very small quantity of zinc channels into my roots. And that way I won't take in too much zinc and I won't get poisoned. So that's passive exclusion. Active exclusion is essentially coping when poison ions do get in, because every once in a while something will. So a lot of these toxic ions are going to be metals. Metals are oftentimes bad for plants. And so one mechanism of active exclusion is going to be through something called metallothioneins, and phytochelatins. So we have metallothioneins and phytochelatins, and these two molecules can both bind to those metal ions to essentially neutralize them and stop their effect. The other method is we're going to have in the tonoplast membrane, which you can think of the vacuole membrane. So remember, vacuoles are storage units inside of plant cells. So on that membrane, there can be a proton sodium antiporter that can force sodium into these large vacuoles and just keep them there and basically go, the sodium can't hurt me if I wrap it up in this blanket of my vacuole. And so that's going to be facilitated through the proton sodium antiporter. And we remember, we talked about the cotransporter up here, or the symporter is another word for this cotransport. So symporter is when things are going in the same direction, because this hydrogen was going into the cell and this chloride was going into the cell. Antiport just means one thing is going to go in and one thing is going to go out. So I have my membrane here, and this is the membrane separating the vacuole and the cytoplasm. What I can do is I can say, okay, I'm going to push Na plus into the vacuole through this channel, and I'm going to push protons out through this channel. And this is secondary active transport, so we would have had a proton pump right here, pumping the protons in to create the gradient. So that's antiporter. So those are the two methods of active exclusion. We're very close. Last page. All right. So next, the last thing is nitrogen fixing. And nitrogen fixing is a process where we take atmospheric nitrogen, which is N2 gas, which plants can't use, and we're going to convert it into a usable form of nitrogen like ammonium, which is NH4+, or nitrate, which is NO3-. There is also nitrite, which would be NO4-, I think, something like that. You don't need to worry about that, though. So we have this atmospheric nitrogen, which is nitrogen gas, that plants can't uptake. So what can uptake it? Well, it's going to be bacteria, specifically these bacteria called rhizobia. So this bacteria is going to take in this nitrogen gas, and it's going to convert it into these usable forms like nitrate, ammonium, or nitrite using an enzyme called nitrogenase. Hopefully that's easy to remember, nitrogenase. And it's going to convert nitrogen gas into NH3, which can then be used by the plant, or it can be used to synthesize ammonium nitrate. So the reason that this is hard for a plant, the reason that it can't absorb this nitrogen gas, is because nitrogen gas has a triple bond, which is really, really strong, and plants don't have the mechanisms to break those bonds or to uptake the gas. So you need nitrogenase-rich rhizobia bacteria to convert atmospheric nitrogen into a usable form. So this nitrogenase enzyme is going to be very poisonable. It can be killed by oxygen, and that's not good. So we're going to create another mutualism with plant roots. For example, legume roots. Legumes are like peanuts. That's the one I always think of. That's a legume. So these bacteria, these rhizobia bacteria, will combine with legume root hairs to form these things called nodules. And we're going to get into that process down here. And these legume root cells have something called leg hemoglobin kind of like in blood, and leg meaning legume, not like a human leg. So leghemoglobin is going to be able to bind oxygen and take the oxygen out, kind of like our hemoglobin does, and that's going to protect the nitrogenase of these bacteria. So the mutualism is the plant is going to get nitrogen, and the Rhizobia bacteria is safe from oxygen. And so it kind of goes like this. This is how a nodule forms, because a nodule is actually a root that has been infected with Rhizobia. We say infected, but infected doesn't necessarily mean a bad thing. In this case, it's a very good thing. So here we have our root hair, and then we have our roots over here. These root hairs are going to release these substances called flavonoids, and these flavonoids are going to attract Rhizobia. There's a little heart. They're attracted to each other. And this Rhizobia is going to move towards these flavonoids, and once it interacts with the flavonoids and interacts with this root hair, it's going to enter the root hair and create what is called an infection thread. And so now we have a bunch of these Rhizobia bacteria, and they're going to form this sort of channel in the root hair to get into the cortex. Once in the cortex, these bacteria are going to start multiplying like rabbits. So they're just going to keep replicating and replicating. And eventually it's going to swell up this infection thread, and it's going to create this like nub almost that's called the nodule. And now we can fix nitrogen. So this was all regulated by flavonoid release. So the plant, if it releases more flavonoids, it's going to get more nodules. So why would it want to release more flavonoids? Well, it would do that if it wanted more nitrogen. So this whole process is ultimately regulated by nitrogen needs. All right, one last tiny little extra topic that I forgot to write something about is these types of plants that I just want to very briefly mention because I think you probably have a good idea of these words. We have parasitic, epiphytic, which is the one you won't know, and carnivorous. Parasitic are plants that grow on other plants or any organisms. Actually, it could be on animals. And they actively are harming the host. Epiphytic plants grow on larger plants. No harm done. So you might see this like an epiphyte might be growing on the leaves of a tree. These like other tiny little plants on just like on the leaf or on the bark. And then carnivorous plants like your Venus flytrap typically have modified leaves. And they consume living organisms. And these carnivorous plants exhibit something called phenotypic plasticity. I'm going to go more into this in chapter 40. But phenotypic plasticity just means the ability to change phenotype, which is expression. So it's essentially the ability to change the genes that you're expressing or how much you're expressing in response to your needs or the environment. All right, guys, that is it for chapter 37. I will see you in the next one for chapter 38, which I believe is angiosperm reproduction. All right, have a good one.